Okay, first thing I want you guys to start with is writing the cell cycle summary so you guys can talk about it with your group. So go ahead and watch the video and write a quick summary. So real quick write. Go ahead and get started with that. Click play and write your summary. Elijah, can you see me? Can you see this?
I can see the presentation.
Okay, hold on a sec.
I'm watching the video right now. Oh, okay, now I can see you.
Okay, good. All right, go ahead and watch the video.
Yeah.
Put it in perspective.
All right, guys. We should have been able to write our summaries. Let's go ahead and get started. We're going to start with interface. So here's what we're going to do. And if everybody would look at the screen, and Elijah, again, if you can't see something, please let me know. So I got two different cameras going on here so that you can see. Okay, so first thing we're going to do is we're going to grab one color. Let me go get some gloves. Make sure you're wearing gloves. So whoever's doing this needs to be wearing gloves, right? All right, so grab one color. I'm going to grab some gloves too. I'm telling you to, and I should be doing the same thing. Okay. Elijah, can you see my whiteboard?
No, I'm looking at the desk right now.
You're looking at the can you see my hand?
No, I think my screen's frozen.
Your screen is frozen?
Yeah, maybe.
So you don't see me moving?
Mm-mm.
Can you like refresh?
Yes, ma'am.
Or click out and click back in? All right. We ready? We're all ready? Yes? Okay. Did you pick one color? Awesome. Okay, we're going to make our membrane with our color. Okay, so this is the way I like to do it. Roll, roll, get it as thin as possible. Elijah, can you see this now?
Yes, ma'am.
Okay. Yeah, I want you to do it on the board. I'm going to make mine just a little bit bigger. All right. How we doing? You got your cell memory? How we doing? Okay. Now, I'm going to go ahead and make my nucleus right here. So I'm going to start with the nuclear membrane. Pick a different color, please. Guys, Do me a solid. Make sure that you're not mixing up the colors. Okay? All right, here's my nuclear membrane. Okay, now I'm going to switch over to a different color. I'm going to use this color for my chromatin. Now the chromatin at this stage is hard to see, so it kind of just looks like a big glob in the middle. So it's kind of hard to recognize. It's not very well condensed, so I'm just going to put that big glob in the middle there. So that's my chromatin. Okay, something else that's in the nucleus and you need a different color is the nucleolus. Okay, so that's the chromatin. So I'm going to get a different color now. And I'm going to put my nucleolus in there. These things are really fun to open, aren't they? Only they're not. Yes. The purple is my chromatin. Okay. Okay, that's the nucleolus. Now I'm going to get another color. The what? This is the chromatin. This is the, hey, guys, this is the chromatin. Okay, so this is all your DNA protein, right? The chromatin. This one is the nucleolus. Okay, different color. What does the bottom say? Nucleolus. This is a nucleolus. Hey, guys, you need to be listening and less talking. I just said it. This is the chromatin, right? So that's what you label this. And here's your nucleolus. The nucleolus. Right here. This is the nucleolus. I labeled it right there. All right. These right here, these are my centrioles. Centrioles. This whole process is called interphase. Take your picture. All right. The person who is doing the actual Play-Doh is doing the Play-Doh. The other person is writing the description, right? That's the plan. So take out your foldable, take out your notes, whoever's doing that part. You need to be writing everything that happens during interphase. So that is 90% of the cell cycle. Elijah, you can screenshot this. Do you know how to screenshot? Centrioles. No.
Once we take the picture, we can assemble it.
Yeah. Okay. So, guys, have we taken this picture already? All right. We ready to move on? So, interface is done. Listen. Yeah. His Microsoft Teams isn't working now. It's okay. Tell him that I'm recording it so he can see it. That is really stinky. All right. Okay, so after interphase, we go into mitosis. What's the first phase of mitosis? That's the first phase of interphase. Let's do prophase. All right, so first things first. Let's take this nucleolus out. Guys, listen. During prophase, the nucleolus disappears. So bye-bye nucleolus. Take that out. The nuclear membrane starts to break apart. So break up your nuclear membrane. Now the chromatin looks like double-stranded chromosomes. So what's really cool about that, they look like really cool double-stranded chromosomes. So let's make them double-stranded chromosomes. They're nice and condensed. Let's make 4 chromosomes. Now we know we have 46 chromosomes, but nobody here wants to make 46 chromosomes, so we're going to make 4. The centrioles began to get closer to the post, and something new, so you need your last color. We're going to now start seeing the spindle fibers form. So you're going to roll these real thin. Okay, so like I said, one person is writing the description while the other person is playing with Play-Doh. What? It's not the same color, but it looks like it, doesn't it? I used to do, it's a different color, but it just kind of looks the same on the camera. All right. So this is Proface. Take that picture. Okay, guys. I know you are, but there's a lot of talking. You guys listening? So those of you writing the description, the nuclear membrane, guys, the nuclear membrane starts to dissolve. The nucleolus disappears. The chromosomes are condensed and they're double-stranded because we've already gone through replication during the S phase and interphase. So they're double-stranded. The centrioles are trying to get to the poles, and you can see the spindle fibers forming. And that's what prophase looks like. I'll give you guys a minute. Do you need to catch up?
Yeah.
Okay, guys, as a reminder, Ms. Johnson divided you up so that one person is doing the typing and one person is doing the Play-Doh. So there should be less talking and other stuff because one person is over there typing, looking at their notes, typing stuff up, while the other person's over there listening to Ms. Johnson playing with Play-Doh. By the way, I've got some paper towels over there if you need some paper towels to clean up your whiteboard. Prophase? Spindle fibers? Okay, we good? Guys, can we move on?
Yes.
All right. What comes after prophase? Metaphase. Okay, so let's do that. So I'm going to erase this. And I'm going to erase that. Okay, so during metaphase, the centrioles are going to go and be at the poles. Guys, the centrioles are at the poles north and south of the cell. Okay, we're going to stretch these spindle fibers out as much as we can. And then we know it's metaphase because these double-stranded chromosomes are doing what? They're kind of lined up in the middle. Okay, so let me get my marker and write metaphase. Take that picture. So whoever's doing the typing is describing metaphase, right? All right. What's after metaphase? Anaphase. Okay, so I'm going to erase that and this. I'm going to shorten these spindles here. Just going to shorten them up. And then I'm going to separate the chromosomes into sister chromatids. And like I said, the way I used to remember is they look kind of like letter A's. And that's how I would remember that was anaphase. So sister chromatids are separated. Okay, so it's almost like they're being pulled apart, right? So this, it's like the centrioles are like fishing rods, and they're just reeling them in. Okay, so they're being pulled apart. And that's anaphase. That's anaphase. Take your picture. Okay, so one person is doing the description, right? And the other person is Play-Dohing, which sounds weird, but whatever. What's after anaphase? All right, cool. So I'm going to take that off. Okay. I'm going to lengthen my cell membrane just a little bit. I'm going to get rid of my spindle fibers because I don't need them anymore. No spindle fibers in telophase. And then I'm also going to bring back an oldie but a goodie. My nuclear membrane, it starts coming back. But not only is it coming back for one side, it's also going to come back for the other side. Because now I'm making 2 cells. So I'm going to bring that in. Bring it in. So now I can start seeing my 2 new cells kind of forming. Each new cell has its own set of centrioles. As well as the nucleolus comes back. Okay. Okay. So notice I start to get 2 new cells being formed. My nuclear membrane is coming back, right, in both new cells. Yes? You can see my chromosomes still. They're still condensed. The nucleoli, so the nucleoli in both have reformed. I have centrioles in both, but no spindle fibers. And then that little kind of butt part is called the cleavage furrow. Okay, but the cleavage furrow only occurs if it's an animal cell. So this is telophase. And then the last part would be cytokinesis, right? And so cytokinesis, you just kind of close it in even more.
What does this say, nuclear what?
This is the nuclear membrane. Nuclear membrane. So it's starting to form back. Remember, it disappeared during prophase. Take that last picture. Huh? I think it's great. I was just looking to see if we have one more to do. Guys, cytokinesis looks almost exactly like Tila face. They kind of go hand in hand. And so for that last picture, for cytokinesis, just bring your cleavage furrow in a little bit more. Start getting the circle to form a little bit more. And so this is the end. Yeah, Anna, you guys are behind? You still working on Tila face? Oh, okay. All right, so guys, I need for you to make sure that you've written each other's names, either up here or up here somehow. So I make sure that I give everybody who worked credit. Make sure that your name is on there. Make sure it's turned in. So check the person who's doing the typing to make sure they turned it in. One person can turn it in, but if you don't trust that person, you might want to go ahead and have them share it with you, so you can turn it in too. Hey, beautiful children, do me a solid. Can you make sure to put the Play-Doh back in the right colors, and do not mix Play-Doh? I didn't like that when I was like 4 or 5, and I still don't like mixing Play-Doh. It upsets me. Make sure to segregate out this Play-Doh. And then close it real good so it doesn't dry out. I think we all know that dry Play-Doh is not fun. Who mixed the Play-Doh? Who is it? Tell on him. Somebody mixed the Play-Doh? make sure your Google Slides is turned in correctly, and then please come turn in your materials. Your unmixed Play-Doh, please. Yeah. I know, right? Yeah. All right, next class, we'll talk about baby making. Yes. You already know how it works. We're going to talk about it on the cellular level, not on the physical level. You learned it? Okay. Thank you. Okay, guys, I need my markers back. No homework tonight. So if there's any homework you haven't done, now you have another night to do it. Okay, so make sure you do that. That's a whole nother level. I'm gonna power play. That's important. I know, right? All right, we're good. Don't forget to throw your gloves away. First, you're going to touch the front. On that part, you got to try to tell that. That's the number side one. You know? With the G1, G1, S, G2, and then because you don't have any access to campus at all? Oh, okay, and you don't have your phone. Yeah, I want you to okay. Play Play-Doh. The Play-Doh? Oh, I gotta use it for that. But our next class will be able to. I can play. You need to go. Okay. Okay, so question. Everybody turned in their Google Slides, right? Yes? And you wrote the name of your partner, or that one group that had 3. You wrote all the person's names on there, right? So I can, when I grade it, I know who's whose and who to give credit to, blah, blah. All right. Okay, so I think I can turn this off. Okay. Does that work if I do that? Stop.